Hello guys and welcome back to my YouTube channel. So we are going to be talking about cell structure and everything that I feel like you need to know about cell structure in A-level biology, regardless of what exam board you're doing. I'm sure it applies to all exam boards. So let's get started. And also make sure, just before we get started, just make sure to like and subscribe if you like this and you want more videos like this, because that will be very helpful. Thank you. So the first thing I feel like we need to talk about and you need to know about cell structure, that's in top of cell structure, is magnification and resolution. You need to know the definitions of magnification and resolution. So what I've put down here is that magnification is the number of times larger an image appears compared with the size of the object. You just need to know this. And resolution is the ability to distinguish 2 points on an image. The higher the resolution, the sharper the image. The reason why I actually included that end bit there is because I have seen in some mark schemes when they ask, oh, what's the definition of resolution? And it's normally worth like 2 marks. Just saying the ability to distinguish between 2 points is gonna give you 1 out of 2 marks. What they want you to say is to say that, but also say that it also creates a sharper image. So yes, learn that resolution is the ability to distinguish between 2 points, but also learn that a higher resolution means a sharper image, a fine image, a sharper image, a better image, something like that. If you learn that and get that ingrained in your head, whenever you see that type of question in your exam paper, you'll get those 2 marks. So please do that. So now I'm going to be talking about light microscopes. In terms of light microscopes, I have put the advantages and disadvantages of them because that is very important, and I do know that exam boards will ask these types of questions, so it is a must to know them. At least know a few of them, but it's good to know. So the advantages are that it's relatively cheap, easy to use, it's portable, and you are able to use it like in school. You are using it in school and you can use it in laboratories. That's what it means, is portable, easy to carry. You can view specimens in color, and you can view living specimens, alive specimens. Like, these specimens can be alive or dead, to be honest, but the advantage is that you can view live specimens. So the disadvantages is that it has a low resolution, which is maximum for most, if not all, light microscopes is 1500, and they have a low resolution, which is around 200 nanometers. And it is difficult to see smaller organelles such as ribosome and the Golgi apparatus. Remember though, with a light microscope, you can see organelles, larger organelles like the nucleus and possibly the chloroplast, because they are large organelles. I have seen in a question, I remember doing a question when they were like, what can you see with a light microscope? And they gave you marks for you saying, oh, you can see a nucleus, for an example. Or I think I've also seen a question where it was like, why can you see the nucleus in a light microscope? And the answer to that was basically that the nucleus is the largest organelle. Because it's a large organelle, that's why you can see it under a light microscope. So it's very important for you guys to know that although, yes, you can't see these small organelles like the ribosome, the RER, but you can also see the nucleus and the chloroplast. Well, those are the 2 main things that you can see anyway with a light microscope. So very important for you guys to know that. Now you have the laser scanning, also known as the confocal microscope. So these microscopes have depth selectivity and can focus on structures at different depths within the specimen. And they use laser lights to scan an object point by point, and you can view living specimens, living cells in color. That's the main advantage of it. And sections can be made through the cell, either vertically or horizontally. Not really something that I would say you need to know, but you know, nothing like learning more sort of stuff doesn't really hurt. So, and also a disadvantage is that its magnification and resolution is low compared to electron microscopes, which we will talk about next. Calculating magnification. So first thing you need to know is that the total magnification of a microscope is the magnification of the objective lens times by the magnification of the eyepiece lens. The eyepiece lens magnification is normally like times 10, and the objective lens vary depending on which objective lens you're using, so times 4, times 10, and times 40. And you basically just times those ones together. So if you have a times 4 objective lens with a times 10 eyepiece lens, it's going to equal to a total magnification of times 40. And then you also need to know about magnification is equal to image divided by actual size. So one way that I remember it is through this triangle, which is the I am. I always remember I am. Whenever I'm doing questions like that, I'm always drawing a triangle. I'm always like I am, and that's how I get through it. Now we are on electron microscopes. So that you guys can understand why electron microscopes have a higher resolution and magnification is mainly because electrons have a shorter wavelength than visible light, and I'm sure you guys will remember wavelength. Like wavelength, as soon as you hear wavelength, you should be thinking physics, because I'm sure we all did physics GCSE, and we remember wavelengths, waves, and everything like that. Transverse waves, longitudinal waves. Okay, I'm gonna off topic a little bit, but the reason why electron microscopes have a higher resolution and magnification is because electrons have a shorter wavelength. So transmission electron microscopes, they can only view dead specimens because the specimens have to be dehydrated and stained with a metal salt, and they are put under a vacuum, electrons, and the electrons passes through the specimen. The reason why they are put under a vacuum is because you're using electrons. If you don't put it under a vacuum, then the electrons could the air particles or molecules could kind of like disrupt the electrons, and the electron will just be like bouncing on the air molecules. That makes sense. But yeah, it has to be put under a vacuum. In terms, again, advantages and disadvantages, you need to know them. Advantages of an transmission electron microscope is that it has a high magnification of times 2 million, which is a lot. It has a high resolution of 0.1 nanometers, and it looks at smaller organelles such as the ribosome and the Golgi apparatus. I have seen in mark schemes, it is actually very important for you guys to have an idea of the resolution and the magnification, because I have seen like questions asked what is the resolution of a transmission electron microscope. So it is very important for you guys to just have it at the back of your head that it's around 0.1 nanometers. So very important. And the disadvantages of a transmission electron microscope is that specimens can only be viewed in black and white, so there's no color, black and white, no color at all, and the specimens have to be dead, as I said, they are dehydrated and they are put under a metal salt that I've said before, and they only produce a 2D image of the cell, because in real life these cells are not 2D, they are 3D. So a disadvantage to using a transmission electron microscope is that it produces a 2D image. The obvious ones are that it's larger and is very expensive, and that it requires a lot of skill and training to use. So not just anyone, like not just me who has like no experience or skill in electron microscopes can use it, because I would not be able to know what I'm doing. And maybe you too, like, so it does require a lot of skill, unlike the light microscope. Like, I can use a light microscope, and I'm sure you can use a light microscope. Scanning electron microscopes, the other electron microscope, SEM, whatever you like to call it, and it uses electrons to produce a 3D image of the outside surface of the cells. So SEM, scanning electron microscopes, are actually used to look at the surface structure of the cell in more detail, and the electrons do not actually pass through the specimen, unlike the transmission electron microscope, but the secondary electrons, they bounce off the surface of the specimen. And as I said, for the transmission electron microscopes, it is also put under a vacuum because you're using electrons, so gotta know that. As always, you need to know the advantages and disadvantages of a scanning electron microscope. And advantage is that high magnification. It's important for you guys to know the value, the number, which is times 200,000, and it has a high resolution of about 20 nanometers. And also something that I didn't even include in the advantage is that it produces a 3D image, which I personally would say is an advantage. Yeah. And disadvantages is that you can only view dead specimens, and it's larger, very expensive, and it requires skill and training. So I can't do it because I have no skill or training. and maybe you as well. So quite a lot of skills and training. Yeah. So you need to know how to convert units. As you need to know, you need to know this. So from meters to, when you're going from meters all the way down to nanometers, so meters to millimeters to micrometers to nanometers, you are basically timesing by 1,000. But when you are doing nanometers to micrometers to millimeters to meters, that's when you are dividing by 1,000. Yeah. And also I do know that you can also use like standard form in meters. So in meters, for I think millimeters is times 10 to the minus 3, and then micrometers is times 10 to the minus 6, and then nanometers is times 10 to the minus 9 meters. Obviously they're in meters, so yeah, you just need to know this. So unfortunately, and maybe fortunately, you need to know this. Using a light microscope, I feel like it's important for you guys to have an understanding of how to actually use it, because you may be asked a question on how can student A make a microscope, use a microscope, for an example. So I feel like it is important for you guys to know, have an idea or understand how you use it, and I'm sure you guys have used it in school, so should not be too hard, hopefully anyway. So the specimen on the slide is placed on the stage, and use clips on the stage to just hold the slide down and just to keep it in position. Then you use the lowest power objective lens, which provides a larger field of view, and you adjust the coarse focus knob while looking through the eyepiece so that you can get a clearer image and so that everything's in focus. And I kind of added an extra sort of thing, but you may also adjust the iris diaphragm for optimum light. Making slides, another important thing that I feel like you should know as well. So you take a thin slice of the specimen and place it onto a clean microscope slide. And you add a drop of water or stain onto the specimen. Then you use a clean cover slip and lower it slowly and gently down at an angle. So you're not like lowering it like this. You're like, I'm not sure if you can see, but you're like this, aren't you? You're lowering it down like this. And yeah, you lower it down on the specimen. And also be careful, like try not, make sure that there's no air bubbles in there. Another thing I found actually very interesting, that I also got, is that you can also put like a drop of stain on the side of a slide next to the cover slip and draw the stain under the cover slip by placing a piece of filter paper next to it, which I find very interesting. So, and I have read that I think I have like seen that or read that in some websites and some stuff. I think I have. But yeah, interesting thing to know. Stains. So there are different types of stains. So aseptic or ausin, I'm not sure if I'm saying that right. So that binds to DNA and stains chromosomes dark red. And I'm just gonna say bullet point 2 because I don't want to say it wrong. These stains cytoplasm, Sudan red, stains lipids, iodine in potassium iodide solution, stains cellulose yellow, which is in cellulose, as you may know, is in plant cells. It's the plant cell, the cell wall. It's in the cell wall. Yeah, cell wall, that's it. And they stain starch granules blue-black. I will do a video. I think I will do a video on that kind of topic of biology. And methylene blue is an all-purpose stain, so it stains everything. So what is the purpose of stains? Because I have seen in exams that they've actually asked you what is the point of staining, or they've asked you a sort of similar question like that. So I'm gonna answer it for you. What is the purpose of stains? So since most cells are colorless, stains add a contrast. And stains that bind to different cell structures help to identify the cell structure easily. And this is also known as differential staining. So that is the main purpose of stains. That's why you stain cells. So very important for you guys to know. Using an eyepiece graticule. So microscope eyepiece are fitted with a graticule. They are normally fitted with a graticule, and we use the eyepiece graticule and the stage micrometer, also known as the stage graticule, to measure the eyepiece unit. So a stage graticule / micrometer is used to calibrate the eyepiece graticule. You place a stage graticule onto the microscope stage and use the lowest power objective lens. This is all about how to calibrate the eyepiece, because I feel like this is so important, because I actually used to not be able to understand it. Like when my teacher was teaching me this, I generally did not understand it, and I'm gonna be honest with you. So very important. So first thing you do is you place a stage graticule onto the microscope slide and use the lowest power objective lens, which is normally the ×4. So when you are using the ×4 power objective lens, you have to be aware that the total magnification is ×40, as I have said in my previous slides that, you know, the eyepiece magnification is ×10, so ×4 times by 10 is ×40. And then you have to align the eyepiece graticule with the stage graticule. And remember that the stage vacuole is 1 millimeter long, which is the same as equivalent to 1,000 micrometers. And it is divided into 100 divisions. So each division is equal to 10 micrometers. And then you have to count how many eyepiece vacuole divisions corresponds to a set number of stage vacuoles. And then you have to calculate one eyepiece unit. So how do you do it? So let's say, for an example, 40 eyepiece vacuole divisions is equal to 1,000 micrometers on the stage micrometer. Then that means that 40 is equal to 1,000 micrometers. So to find one eyepiece vacuole division, you basically do 1,000 divided by 40, and that will give you one eyepiece one eyepiece unit is equal to 25 micrometers. Does that make sense? Now we're looking at eukaryotic cells. So as you can see, there is a table of these organelles, organelles that animal cells and plant cells have and do not have. So as we know, they both have a nucleus, nucleolus, rough endoplasmic reticulum, smooth endoplasmic reticulum, ribosomes, mitochondria. Animal cells do not have chloroplasts, plant cells do, and they both have Golgi apparatus. Animal cells have centrioles, but most plant cells do not have centriole. Cellulose cell wall, only a plant cell has that. They both have a plasma membrane. Plant cells have a large centriole vacuole. Animal cells do not have a large central vacuole, although I do know that some animal cells do have a vacuole, but it's not a large permanent one, and they both have a cytoskeleton. Now we are going to be talking about comparing eukaryotic cells with prokaryotic cells. So the first thing we're going to look at is similarities between eukaryotic and prokaryotic cells. So the first thing is that they both have a cytoplasm, they both have a cell membrane, they both have ribosomes. Some eukaryotic cells do have a cell wall, and prokaryotic cells have a cell wall. So eukaryotic cells like fungi, why did I say fungi? Fungi and plant cells, they have a cell wall. They both have a cytoskeleton, although prokaryotic cells' cytoskeleton is more simpler. And they both have DNA and RNA. Differences, and when I'm saying the differences, like, take note of like how I'm describing the differences, like the structure I'm using, because that is really important. So prokaryotic cells have a cell wall made up of peptidoglycan, whereas eukaryotic cells have a cell wall made of different materials like cellulose in plant cells and chitin in fungi. I don't think I'm saying it right, but who cares? Prokaryotic cells, they are smaller than eukaryotic cells. Prokaryotic cells have ribosomes of size 70S, whereas eukaryotic cells have ribosomes of around 80S. So that means that prokaryotic cells, their ribosomes are smaller than eukaryotic cells' ribosomes. Eukaryotic cells have membrane-bound organelles, such as mitochondria and the nucleus, whereas prokaryotic cells, they do not have membrane-bound organelles, so they do not have a nucleus or mitochondria or any other organelle that is membrane-bound. Prokaryotic cells have a waxy capsule, whereas eukaryotic cells do not have a waxy capsule. DNA in eukaryotic cells are found in the nucleus, whereas DNA in prokaryotic cells are found free in the cytoplasm. Inside the nucleus of the eukaryotic cells, the DNA is wrapped around histone proteins, whereas in prokaryotic cells, DNA is naked. DNA in prokaryotic cell is circular, whereas DNA in eukaryotic cells are linear. Prokaryotic cells have plasmid DNA, whereas eukaryotic cells do not have plasmid DNA. So, from that, you should have seen that I said, instead of just saying, oh, prokaryotic cells are circular. No, let's not even use that example. Oh, instead of just saying, oh, eukaryotic cells have membrane-bound organelles, I didn't just say that. I said, I compared. So I said that, yes, eukaryotic cells do have membrane-bound organelles, but prokaryotic cells do not have membrane-bound organelles. Do you know what I mean? When you are answering these questions where it's like, oh, state the differences between prokaryotic cells and eukaryotic cells, don't just say, oh, eukaryotic cells, oh, they have a nucleus, they have membrane-bound organelles. No, you should say that eukaryotic cells have membrane-bound organelles, whereas prokaryotic cells do not, or however, but you need to use those types of structure, and that is really going to help you, because I have done it before. Like, I have done it in my test where I've just said, oh, eukaryotic cells, for an example, they only have a membrane-bound organelle. Even though it's right, I'm not comparing, I'm not contrasting. That's what you need to do in your exam. So whenever you are comparing between eukaryotic and prokaryotic, or any sort of thing, when you are comparing two things, always say this has that, but that does not have this. You know what I mean? So yeah. The nucleus. So the nucleus contains DNA, which is genetic information, and chromatin is a genetic material which consists of DNA wound around histone proteins. So the nucleus is surrounded by a double membrane, also known as a nuclear envelope, and the nuclear envelope contains pores, which are basically like holes, known as nuclear pores. And the main function of the nuclear envelope is to control what enters and exits the nucleus. So for example, mRNA exits the nucleus, and some steroid hormones may did I say enters the nucleus? No. mRNA, it exits the nucleus, and some steroid hormones may enter the nucleus. The nucleolus is where ribosomes are made, and what am I saying? The nucleus provides instructions for protein synthesis, transcription, which I will talk about in another video, so stay tuned for that. So as you can see, I have labeled, for example, in bold. The reason why I've done that is because, again, I have seen questions where it was like, if I remember correctly, it was like, what is the function of the nuclear envelope? And it was 2 marks. And in the mark scheme, they said, not only should you say, oh, it controls what exit and enters the nucleus, they also gave you a mark for just saying an example, saying how, oh, mRNA needs to exit the nucleus, or steroid hormones enter the nucleus. Like, just by giving examples, they gave you a mark. So it's important for you to know and understand the function of the nuclear envelope, and also give examples as well, because that is going to force the examiner to give you a mark. The rough endoplasmic reticulum, which is also, you know, if you want to abbreviate it, you can say RUFIA or ARIA. So that provides a large surface area for ribosomes. It contains ribosomes and it transports proteins made by ribosomes. That's really all I can say about them. The smooth endoplasmic reticulum, the SER, smooth ER, whatever you like to call it. This is for lipid synthesis, synthesis of steroid hormones, synthesis of cholesterol, and it's involved with absorption, synthesis, and transport of lipids, as you can tell. So the Golgi apparatus, it modifies proteins and packages them into vesicles. That is their main function. So they can modify proteins by adding carbohydrate chains to make glycoproteins, modify proteins by adding lipids to make glycolipids, and they can also modify proteins by folding them into their 3D structure shape, tertiary structure. You will know more about it in the protein side of biology, and I will hopefully do a video on that, so stay tuned. So the mitochondria is the site of aerobic respiration, as all of you should know from GCSEs. Actually, I'm going to take off my glasses. Ooh. Sorry. As all of you guys may know from GCSEs, we know that it's a site of aerobic respiration, but it's also the site of the production of ATP. It is spherical or rod shaped. So if you are looking at a graph, at a picture of the mitochondria, it will either be spherical in shape or rod shaped, depending on how they cut the cell. So whether they do it longitudinal or the other way, again, it depends. It's this double membrane, so it's surrounded by two membranes. The inner membrane forms a cisternae, so that's the one that's like this. Oh, it's like this, as you may see from pictures. And the inner part of the mitochondria is filled with a fluid film matrix, and the mitochondria is self-replicating, so it contains DNA. So in terms of the inner membrane, defaults was like a cristae, if I can. So you would kind of see like a sort of shape like this, or if you can see it, something like that. Yeah. So chloroplast is around 4 to 10 micrometers long, and it is the site of photosynthesis. It is surrounded by a double membrane, and the inner membrane has stacks of flattened sacs called vacuoles, which contains chlorophyll. And each stack of the vacuole is a granum. So again, so it will kind of like look something like, ooh, oh, this is not a very good representation, but you will kind of like see stacks of it. So this stack is a granum, but each of the stack is a vacuole. So this is not even a good representation, but I'm sure if you search up pictures and you see like the diagram, you will understand what it means. And the stroma is a fluid filled matrix, and the chloroplast has its own DNA, and also the mitochondria also has its own DNA, so it is self-replicating. Not sure if I said that in my mitochondria slide, but that's for you to know. So the vacuole, plant cells only have a large permanent vacuole, but plant cells contain a large permanent vacuole. You may see in some animal cells that they do have a vacuole, but it's not a large and permanent one. They're different. A vacuole, the main function is it maintains cell stability and it contains water and solutes. So when a plant is turgid, when there's a lot of water, there's excess water like entering the cell by osmosis. Again, I will do a video on that. The vacuole just supports the plant, and the membrane of the vacuole is called the tonoplast. Lysosomes, these contain hydrolytic enzymes, and these isolate the hydrolytic enzymes from the rest of the cell, because these hydrolytic enzymes will break down waste materials and will break down cell. If there weren't lysosomes that were isolating the hydrolytic enzymes, then these hydrolytic enzymes will digest like all of the organelles in the cell and there'll be no cell. So the lysosomes are very important, and they are found, a lot of them are found in like white blood cells, especially phagocytes. They contain so many lysosomes, and they contain them to engulf pathogens for the immune system. Cilia. So cilia, the main function is movement of cells on substances. You will know cilia from, if you've heard of the ciliated epithelial cell, where their function is they use the cilia to like create a sort of current, would you say, and they move mucus which has trapped dust or microorganisms away from the lungs up the throat so that it can be swallowed and go towards the stomach. Yeah. Undulopodia, which is basically the tail that you see on a sperm cell, and that their main function is the movement of the cell to move the cell. As you know, in the sperm cell, there needs to be a there needs to be a tail so that it can move. That's what I'm trying to say. Undulipodia. Interesting. Hope I'm saying that right. So ribosomes is the site of protein synthesis, and it's about 20 nanometers in diameter in eukaryotic cells. And I believe, I'm not sure about this, but I believe it's around 18 nanometers in prokaryotic cells. I believe. I think so. I think. So centrioles are made out of two bundles of microtubules at right angles to each other, and centrioles are involved in forming the undulopodia and the cilia, and is usually not present in plants. So if you remember in my previous slide where I had the tick 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 on what is present, if you remember, centrioles are not usually present in plant cells, so I put a cross in it. Cellulose cell wall. So this is found in plant cells, and they strengthen and support the plant cell. It maintains the cell's shape, and it is permeable, so they can allow solutes and solvents, solutions to enter and exit. The production of proteins. So each gene in DNA codes for a particular protein, as you may know. mRNA makes a copy of the gene and moves out of the nucleus through the nuclear pores in the nuclear envelope. So remember, the nuclear pores are in the nuclear envelope, and the nuclear envelope controls what enters and exits the nucleus. And this mRNA attaches to the ribosome on the area, known as the rough endoplasmic reticulum, as you may know from the previous slides. The ribosome translates the mRNA, protein synthesis, and produces a protein synthesis, which travels through the area, rough endoplasmic reticulum, and is packaged into a vesicle. So once this protein is made through protein synthesis in the ribosome, it travels through the RER and is packaged into a vesicle. So then this vesicle then travels to the Golgi apparatus, and this is the production of proteins. It's almost like it's the abbreviated version. But I will do a video on protein synthesis in more detail where we talk about transcription and translation. And a question that I actually have for you that you can think of is that I've typed down here is, what is aiding the transport of the vesicle from the RER to the Golgi apparatus? So basically, how does the vesicle have the ability to move from the RER to the Golgi apparatus? So that's what I'm asking you, and it's something for you to think about. So, secretion of proteins. So the Golgi apparatus modifies the protein and then packages them into vesicles. So when we talk about modifying the protein, this could be by adding carbohydrate chains, this could be by adding lipids, or usually is just by folding the protein so they can form its 3D shape, tertiary or quaternary shape. And this is then, once it has been fully modified, it is then packaged into a vesicle. And the vesicle then transports this newly modified protein, to the plasma membrane. Again, how is the vesicle able to move from the Golgi apparatus to the plasma membrane? It's not doing it by itself. There is something helping the vesicle to move from the Golgi apparatus to the plasma membrane. So that is, again, something that you should think about. The vesicle fuses with the plasma membrane, and the protein is secreted outside of the cell through the process called exocytosis. This exocytosis is a type of bulk transport. Again, I will do a video on that. But that's all you should know. The important thing is that you should say what the Golgi apparatus does. It modifies and it packages them into vesicles, which is practically their function. You should say where the vesicle is transporting the protein to, and very, very important, so important, you need to use the word fuses. You need to use that word. You need to say that the vesicle fuses with the plasma membrane. That will give you the mark. And then they may also give you a mark for just saying this process is called exocytosis. I'm sure they will give you a mark for just saying exocytosis. So very important for you guys to do. So the answer to the question that I asked you guys was the cytoskeleton. So the cytoskeleton supports this cell and gives the cell its shape and structure. So the cytoskeleton is basically just a network of fibers that just run through the body. And there are 3 types of fibers in the cytoskeleton: microfilaments, microtubules, and intermediate filaments. Cytoskeleton is used for transport. The answer to my question that I asked you guys. So proteins called microtubule motors, they move up and down the cytoskeleton, the microtubule, and carry vesicles that are carrying proteins for transport, for the secretion of proteins, production of proteins, them stuff. The cytoskeleton is also used for cell movement. The cytoskeleton is used to form cilia and undulopodia. And undulopodia. Is it undulopodia? Yeah, undulopodia. I had to ask myself that. So the microtubule extends out of the cell's plasma membrane to form cilia or undulopodia. These are both made from 9 microtubules arranged to form a circle with 2 microtubules at the center. So it has like a 9 plus 2 type of arrangement. So there's basically 9, what do you say, pairs of microtubules like forming the circle, and then there's like 2 microtubules in the middle. Yeah. Sorry, I was just talking to myself a little bit. So, as from previous slide, I did talk about eukaryotic cells and prokaryotic cells, but I wanted to kind of talk a little bit more about prokaryotic cells a bit more, so that you can understand it and know the other features that prokaryotic cells do have, and kind of explain it in more detail. So prokaryotic cells have a protective waxy capsule, as I said before, and that surrounds the cell wall. The capsule protects the cell from being engulfed by white blood cells of the immune system by hiding their antigens. The waxy capsule allows the prokaryotic cells to stick together, which allows them to share nutrients and genetic material. Being able to stick together increases the likelihood of surviving. I'm sure some of you, maybe if you did know it, but I'm sure some of you may not have known this, so I feel like it's useful information to know. Pili help the bacteria, the prokaryotic cell, to stick together. Pili are hair-like structures which are also made of protein, and these extend out of the bacteria prokaryotic cell. Mesosomes are the infoldings of the cell membrane of the prokaryotic cells, and they basically have a similar role to mitochondria in eukaryotic cells, which is the site of aerobic respiration. Prokaryotic cells, as I had said before, they have a simple cytoskeleton compared to eukaryotic cells, and they have a flagella for cell movement. Thank you for watching, and I hope you guys really enjoy it. This is a very long, but I hope you guys enjoy it and found it useful. Make sure to subscribe if you want more videos like this, and see you in another video. Bye.